Hello everyone, I'm Khalique Newaz from the University of Notre Dame. Today I'll present a work on supervised prediction of aging-related genes. Aging is related to many diseases such as diabetes, cancer, Alzheimer's, etc. Hence, understanding molecular mechanisms behind aging can also help us understand these diseases. Using wet lab experiments to study aging can be time-consuming and costly. Hence, computational approaches are used which are much cheaper and faster. The goal of our study is to computationally predict aging-related genes. Computational approaches to predict aging-related genes can be grouped into three groups. One group of approaches are the approaches that only use gene expression data. However, such approaches ignore the important interaction information between the gene products that are proteins. Second group of approaches only use the PPI network data. However, such approaches ignore the aging-specific gene expression data. Hence, recent approaches integrate the two data types to first create an age-specific PPI subnetwork for each age. Then, because aging is a dynamic process, they treat the collection of age-specific PPI subnetworks as a dynamic aging-related PPI subnetwork. Finally, they use the dynamic aging-related PPI subnetwork to predict aging-related genes. However, all of these approaches use the concept of induced approach to create an age-specific PPI subnetwork. The induced approach selects an induced subgraph of active genes from the whole PPI network to create an age-specific PPI subnetwork. As such, this approach ignores any inactive genes that might be critically connecting active genes in the PPI network, and it also fails to give age-specific weights to the PPIs, which might be important to distinguish between important age-specific PPI from unimportant age-specific PPIs. In this context, there is a concept of network propagation, which can help. Network propagation first propagates gene activities to the entire PPI network and assign age-specific weights to the PPIs. In this way, NP or network propagation is able to include inactive genes. For example, when the inactive gene is connected by many active genes. And also, NP gives age-specific weights to the PPIs. In this study, we hypothesized that creating dynamic aging-related PPI subnetwork using network propagation can better capture the aging-related information than either the induced approach or which gives an unweighted dynamic aging-specific subnetwork or a static aging-specific subnetwork or the entire context-unspecific network. Hence, we test three hypotheses. One, whether aging-specific subnetwork outperforms context-unspecific network. Second, whether dynamic information helps, i.e. dynamic subnetwork outperforms the static aging-specific subnetwork. And third, whether weights help, i.e. whether weighted dynamic aging-specific subnetwork outperforms the unweighted dynamic aging-specific subnetwork. To evaluate these hypotheses, we use a supervised framework in which the input is a PPI subnetwork and a set of aging-related genes. And then we use these input to learn a classification model, which can then be used to predict whether a gene with unknown label is aging-related or not. We evaluate our framework using 5-fold cross-validation, where we divide data into 5 folds and use each fold individually as a test data and the rest of the 4 folds as training data. We quantify the performance using 4 measures that are precision, recall, area under precision recall, and F-score. We find that all of our three hypotheses hold. That is, aging-specific subnetwork is better than context-unspecific. Dynamic aging-specific subnetwork is better than static aging-specific subnetwork. And weighted dynamic aging-specific subnetwork is better than unweighted dynamic aging-specific subnetwork. The two main conclusions of the study are that aging-specific subnetworks always performs better than context-unspecific subnetwork, and weighted dynamic aging-specific performs better than unweighted dynamic and also static counterparts. With that, I would like to thank all of my coworkers and the funding, and thank you for listening.